Okay, why don't we get started? So a couple of announcements before we get started. I told you about the weekly exercise classes that I lead. I just want to say that we're doing a big kickoff event this Sunday. This is the second Sunday class that we've done, and this is a big kickoff event for our Empowering Women in Science explorations floor. Both men and women are certainly invited. It's going to be a workout, but the cool thing is that we're going to have live drummers there. So if you want to come and experience the workout, this is a really good week to come. Sunday from 1:30 to 2:30. You could also sign up for the kind of leadership training overview that we're going to be doing afterwards. And I'm just going to leave this up here at the front. So if you're interested, sign up. Tell me which components you're interested in. You can come to the workout only, you can come to the leadership training only, or you can come to both. Let's see. Is everybody getting all the lectures and the questions off of Blackboard okay? Is that working? Yes.
Some of the documents are in .x and the file for.
Okay. Okay, I could convert those to PDF files. Okay, great. Any other problems? Okay, so definitely download all those files and all the questions and all the information that's been put up there. Okay. Oh, and then this week is your first week of lab sessions. So on tomorrow and Friday will be your first lab sessions, and you will be taking lab with Eric over here, whose lab sessions are?
Thursday at 3 and 5.
Thursday at 3 and 5. Helena up there, whose lab sessions are? Sorry? 9 and 5. On Friday? On Friday. Helena, wave over there. And then Sarah? 11 and 1 on Thursday. Okay, great. So first session, go there. We're going to be talking about scientific method. It's going to be a really fun lab. So that will be this week. So what I wanted to do is hear from you a little bit in the beginning of this class. And specifically, I wanted to ask you guys the question, why did you decide to take this class? What is interesting to you about neuroscience? And it could be from something you've been interested in the past, or it could be something you became interested in the first couple of lectures. But I'd really like to know. So what I'd like to do is split up into groups of about 5. Just naturally go and find about 5 different people and talk about this amongst yourselves for about 3 minutes. And I want to kind of hear from you guys about what interests you in neuroscience kind of at the beginning of this class, okay? So please just introduce yourselves to 5 people around you and tell each other why you took this class. Okay, everybody should be talking or listening right now. Okay. Okay, it looks like you guys conveyed your information. Who wants to share? So give me some reasons why you took this class. In the back. You. Okay, great. She what? Okay. Any other reason you chose this class? I see. Psychology major, so you're interested in neuroscience. Great. Okay. Anybody have like a thing that they became interested in neuroscience that made them want to learn a little bit more about neuroscience? Yes.
I took sensation and perception last semester, which is a psychology class.
Mm-hmm.
And I kind of focused on neuroscience and how we perceive things and what causes that in the brain. So this was like an extension of that into behavior.
Uh-huh.
Neuroscience became my
Great, great. So got interested in neuroscience through her course, Perception and Sensation. Is that? Yeah. So that's great. Anybody else? Unique reasons. How about you? Yes.
Oh, well, I do need it for my natural science requirement.
Okay, need it for your natural science requirement.
But actually, I kind of became kind of interested because my sister did some research into primate psychology.
Mm-hmm.
Told me about it. And yeah, it's really interesting.
Okay, primate psychology. Yes, that's a topic that's really interested me. On my last sabbatical, which was almost 7 years ago now, I had the privilege of going to Botswana to study baboons in the wild, to look to see exactly, what are they doing out in the wild? What kinds of things are they learning? What kinds of things are they remembering? And that was a fascinating experience. They are not doing what you might think they're doing from the tests that are typically done in laboratories, which is exactly why I went out there. But not that they're doing nothing. They are so focused on social interactions. They're primates just like us, and they are interested in who's dominant to who is going to be nice to me, and who is going to beat me up and take my food away. And that is what they focus their cognitive attention on. And because of that, kind of the tasks that I've developed in my lab to test primates focus on memory for those kinds of social interactions. So yeah, that's a topic very interesting to me as well. Anybody else? Yes?
I was really interested in music and memory and how Alzheimer's patients, we work most of their memory on.
Yeah.
A song from like when they were 14, I really remember I still recall
Uh-huh.
memories and how music like sends off sensations in the brain, same as like cocaine and chocolate.
Mm-hmm. Mm-hmm.
And just like how auditory and
Okay, great. Interested in music and the brain and Alzheimer's disease and how music can set off lots of different reactions. One of the most interesting fields of study is the neurobiology of music. And that is really centered at University of Montreal and McGill University up in Montreal. And researchers up there, including Robert Zatorre, have done a lot of work looking at functional magnetic resonance imaging. Do you guys know what functional magnetic resonance imaging is? Yes? No? Yes? Okay, so functional magnetic resonance imaging is using an MRI magnet. Everybody knows what an MRI magnet is. You usually go in there if you have a knee problem, or you can image different parts of the body, including the brain. But functional magnetic resonance imaging, functional MRI, is where you can go in and actually look at an indirect measure of brain activity. And so you can put somebody in the magnet and deduce which part of the brain is active as they're listening to music. And I love the study that Robert Zatorre did on those pieces of music that kind of give you goosebumps. So what the evocative power of music and what parts of the brain are being stimulated when you hear that piece of music, which is different for everybody. Some people don't have any pieces of music that will give them kind of goosebumps because it's so evocative. But some of us have lots of different kinds of music. So he found that piece of music for each person, and there are specific brain areas, including areas involved in emotional processing that we're going to be talking about, including the amygdala, that get activated in these kinds of processes. So it's a new area, but very, very fascinating. We're going to talk a little bit about that in both the auditory part of the class as well as the emotion part of the class. Anybody else? Yes?
I'm interested in dreams. And
Dreams.
Sleep and how sleep affects the brain.
Yeah.
And also what triggers memory, such as like smell.
Yeah.
And what's going on.
Yeah, yeah. So he's interested in sleep and dreams and how smell evokes memory. And the evocation of memory by smell is fascinating because we won't go over this in great detail, but we will talk about the major senses like vision and audition and somatosensation that have specific pathways into the cerebral cortex. They go through the brainstem, they go into the thalamus, and then they go into the cortex. That is the prototypical sensory pathway into the brain. However, olfaction is different. Olfaction has a direct route directly into the brain, does not go into the thalamus. And one of the areas that it goes into is an area that is a major feeder to the hippocampus, the area that we talked about is important for long-term memory, which is why smell can be so evocative for our long-term memories. Sleep is very fascinating as well. A researcher at UC Berkeley has started kind of revivified the study of sleep and memory and showed that even small naps can significantly improve both your procedural memory, your ability to do kind of motor learning tasks, as well as your episodic memory, your hippocampal dependent memory. So there may be a reason why you get that lull and that sleep desire for sleep in the afternoon. You might need something, that extra boost, to improve your memory. And we certainly know that if you I teach this regularly, but it is striking to me that if you deprive, in an experimental animal situation, if you deprive a rodent of sleep, do you know what happens? They die. Sleep is a basic physiological need. It's not like, oh, if I have time, I'll sleep. You have to sleep. And not only do you have to sleep, but it's doing good stuff. It's extra naps, and if you enhance certain stages of sleep, you can enhance certain forms of memory, and that's exactly what's being studied right now. So yeah, very interesting topic. One more. Anybody else? Question or interest? Yes? Déjà vu. Okay. Interested in déjà vu. So that is very interesting. It hasn't been studied a lot directly, at least the neuroscience of it. I think the psychology of déjà vu has been studied more. But certainly what we know is that it involves the retrieval of memory, and that involves the hippocampus. We'll learn that while there's weeks and weeks and weeks of information to learn about neuroscience, we're still at some levels at a very basic level of understanding. So you think, oh, well, we can understand déjà vu, and we can understand how sleep, and we have little snippets into that. So that's one of the things I want to impress upon you. We know an enormous amount, but there's also an enormous amount that we have to learn. And so trying to separate out where we are on very strong ground in what we know, and where we're kind of still searching and asking the experimental questions. Okay, great. So why don't we get started with the main lecture? Today's lecture focuses on neurons and glia, the main building blocks of the brain. So as I mentioned, I believe in the first lecture, the entire human brain, the cortex, the cerebellum, the brainstem, the entire central and peripheral nervous system are made up of simply two types of neurons, cells. Neurons, major brain cells that we consider the workhorses of the brain, and glia cells. There are fewer categories of glia, and I have to say that I think in the next 10 years, it's going to be the next 10 years of glia cells and really understanding and appreciating what these glia cells do. We're going to go a little bit into what we know, but actually that is a very, very exciting area of research. We've ignored them for many, many years because there's so many fascinating things that neurons do. So we're going to really focus the whole course on neurons, but just know that glia, I think, are unsung heroes in this kind of two-stage cell makeup of the brain. Okay, so all neurons and glia are basic cells. From basic biology, all cells have similar makeup. They all have a cell membrane. They all have a cell body. They have nuclear membrane, that is a membrane around the nucleus, a nucleus with DNA that has the codes for all the proteins that are made by the cell. They have mitochondria, the the powerhouse of the cell, and ribosomes, which are structures that help in the production of proteins. So all cells in the body, including neurons and glia, have all of these characteristics in common. So all neurons are similar in these ways to liver cells or colon cells or stomach lining cells. But what we want to focus on today is what's so special about neurons in particular. And here I'm specific about neurons, not as much about glia. What's special about the neurons? One is they have very unique morphology. So neurons have a cell body, like all other cells, but they have two major specializations. One are dendrites, which are the input structures of the cell. And the second specialization is the axon, the output structure of the cell. Okay? The second major specialization of the neuron is the way they communicate. Liver cells just sit there. They do their job in processing different food groups that you bring in. Neurons are special because they communicate with a combination of electrical and chemical synapses. And that communication ability is the basis of how we're able to see, hear, perceive, remember, think, and kind of the basis of our personality. So we're going to be focusing on the next two lectures on electrical and chemical synapses. But today we're going to focus on the basic building blocks, so what makes neurons unique. Okay, so to understand certainly our understanding of the neuron, we need to go back in history and go back to these two figures that I mentioned in the very first lecture. Ramon y Cajal, Santiago Ramon y Cajal, a Spanish neuroscientist that's considered the father of modern neuroscience, and Camillo Golgi, an Italian early neuroscientist. In 1906, both of these two scientists together were awarded the Nobel Prize for, one, the discovery of the Golgi staining technique that we talked about, and two, his use of the Golgi staining technique to really distill out some principles of organization that we now know are completely true way back in the late 1800s. Okay, so Ramon y Cajal was a very, very interesting character. And for those of you interested in history, I would highly recommend this book, Recollections of My Life. It is an autobiography written and recently republished of the life of Santiago Ramon y Cajal. It's great that we have this record of his life because without that we have just pictures. I love this picture because it conveys the joy of parenthood, doesn't it? Just the joy. Look at that. If we only have this picture, we might have certain ideas about Ramon y Cajal. It turns out he was a very devoted parent. He had 6 kids, and he loved his children, even though he doesn't show it very, very generously in the photos. You will not be surprised to hear, after we go through a few more of these slides, that he started out with aspirations to be an artist. And he might have been one of the greatest Spanish artists. but his father said that is just not serious enough. You have to do medicine. So because of his father, he went into medicine. And what he did is combined both his amazing artistic abilities with what turned out to be an amazing scientific mind as well. And that allowed us and allowed him to make some of the first major discoveries in neuroscience and neuroanatomical organization of the brain. Okay, that's Ramon y Cajal. Camillo Golgi was a physician, a doctor, a professor in Italy. I love the title that he had. His title was Extraordinary Professor of Histology. It's like, I wish NYU would give that title, Extraordinary Professor of Neuroscience. No, I'm just professor. But he was Extraordinary Professor of Histology at the University of Pavia. And he was known as an amazing teacher of scientists. So his lab was open. Scientists came to learn how to do the craft of experimental design in his lab. In addition to his development of this Golgi staining technique, which he was most famous for, he was also had his kind of irons in lots of different fires, making major discoveries about malaria and differentiating different forms of malaria and showing the different stages of malaria through histological examination. The focus of his time was the nervous system. Right at the end of the 1800s, early 1900s, people were very interested in the nervous system. So he jumped into science interested in that. Still got into malaria research, was a great teacher, but again, he was the one that discovered how to stain the brain so that you can look at individual brain cells. Now, I told you what this Golgi stain was, and I showed you drawings of the Golgi stain. Here are two pictures of what the Golgi stain really looks like. We use it today. You can buy a kit for the fast Golgi technique today based on the original technique that Golgi used. Of course, it's all made very easy to do. But here you can see a cell body of a neuron and lots of these dendrites coming out. And here you see some of the extra staining of other cells that can kind of engulf a particular brain area and make it completely black if it stains all of the brain tissue in that area. Here's another nice example of what's called a pyramidal shaped neuron. And we see the dendrites coming out here. This is probably this thinner structure right here is probably the axon. But you can see that in this section, we had random staining. Again, random. We can't tell which brain areas in this chunk of tissue or this chunk of tissue is going to be revealed. And it's a little bit luck of the draw, and you just have to stain and stain and stain. But this is exactly what it looks like. A little bit messy. But it was both the scientific mind and the artistry of both Golgi and Cajal that made those drawings that we talked about, that we saw in the first lecture. Now, it's fine to make the drawings, but what is the organization of this brain structure? We know what the organization of the liver was, the heart. How are these brain areas organized? And this is where Golgi and Cajal ended up in a huge fight. In fact, they were not speaking to each other when they came and gave their Nobel lectures and gave their lectures on two very, very different opposing topics. Golgi was a champion of what's called the neuron doctrine. Cajal said that, so this doctrine states that neurons are independent functional and physiological units in the brain, so that each one of these neurons are independent neurons, independent units, like an independent liver cell is independent from each other liver cell. Golgi, looking at the same material, stained by his own technique, looked at the material and he said, wait a second. I support what they call the reticular theory. Reticular theory states that all neurons are linked to one another by anastomosis, which means a continuity. So yeah, there are these long processes here, but they definitely saw that the processes are meeting each other. They couldn't see exactly whether they were continuous or they were separate. It was just beyond the resolution of the microscopes at that time. And Golgi reasoned that this is brain tissue. We know that this is the tissue that is processing our highest level of intelligence and cognition and perception. And so what it makes more sense if there is perhaps a slightly different organization from other boring structures like the liver that doesn't have to do much computation. And he thought that by having more continuous integration, you would have more power to compute. Okay? Golgi said, sorry, Cajal said, no, this is a basic unit. The cell theory, extension of the cell theory, says that each cell is independent. This is no different from the brain. And I believe, even though he couldn't see it for sure, that these individual neurons are independent units. Okay, so as I said, they couldn't resolve this question back then in the late 1800s, early 1900s. And in fact, if we only had the light microscope, even the most powerful light microscopes we have today, still couldn't resolve it. Because, in fact, the technology for the light microscope has been unchanged for over 150 years. And the absolute limit is about 1,000 times, which is simply not high enough to resolve what exactly is happening at the connection from the axon of one neuron to, the axon to the dendrite. Is it continuous or is it not continuous? That was the big question. So here are some examples of Cajal's drawings of Golgi stained material so you can appreciate his artistic abilities. Again, he never saw this beautiful picture. He saw individual stained cells, and he put them together with where they were in the section relative to the top of the cell, and just an accurate drawing of what the different branches look like. Here's a drawing of the hippocampus, a rodent hippocampus, showing the different types of neurons. And here also, he's showing these as individual units, and he's showing the flow of information. He had no idea which flow of information. He wasn't recording these cells. He was just looking at these pictures. But through this look, this study of all the different brain structures, he gleaned, so this is the cerebellum. And here's an individual cell from the cerebellum. He gleaned two major principles that you need to know. One is the law of dynamic polarization. We mentioned this in the very first lecture. The law of dynamic polarization states that information flows in a predictable and consistent direction within each nerve cell from the dendrites, many more dendrites in general in neurons, through the cell body and down to the axon. If you remember, we talked about this beautiful drawing, again by Cajal, of the retina. And you can start to see his logic of thinking here. The retina, we know, is a structure important for vision. And he noticed that the dendrites tended to be shorter, and the axons were the ones flowing out of the retina. The eye is made to bring sensory information into the brain, given that all the cells were facing in the same direction. And these cells were modifications of the cells that you see in the cerebral cortex. He reasoned that all these cells always have a specific flow of information from the dendrites to the axons. Again, no physiological evidence, just from anatomy. Second, principle of connectional specificity. And this is a principle that really just flies in the face of Golgi's reticular theory. Here, in Cajal's principle of connectional specificity, he says there's no cytoplasmic continuity between the nerve cells. In other words, Golgi was wrong. Nerve cells do not form random networks. He said that that's an inefficient way to be able to process information. Instead, given the beauty that he was seeing and specificity of the different brain types and the flow of information. He said that each cell forms specific and precise connections, making contact with only some nerve cells and not with others. He couldn't be sure about this, but again, he gleaned this from his study of the basic, many different brain areas. And it turns out that all of these principles were correct. In the foreword of this book, a very famous neuroanatomist, Max Cowan, makes the comment that all the neuroanatomists that came after Cajal would get really worried if they found something that was different from what he had put in his major papers and books that he published over the years, even though they were published so long ago. Basically, we're using similar techniques. I talked to you about Brainbow and the GFP approaches, but you're still looking at anatomy. And Max Cowan makes the comment that neuroanatomists, if they happen to find something that seemed to disagree with Cajal, got very, very nervous because he was almost never wrong. So you want to make sure you're seeing something, and you kind of, neuroanatomists, even today, will go back and double check to see whether it's consistent with some of Cajal's early observations. Of course, he didn't use everything, and there are certain techniques that Cajal just wasn't able to look at all. But he had that level of precision in his analysis. Okay, so back in when Cajal and Golgi won the Nobel Prize, this debate was still raging. I have to say that the descriptions of the time say that Cajal was winning. That more scientists believe that the neurons were individual units. But again, nobody could tell the difference. And the question is, how was this debate resolved? Okay? We're back in 1907 in the Nobel Prize winning year. It wasn't resolved until the 1950s with the advent of the electron microscope. The electron microscope is a microscope that allows you to go down to a much higher level of resolution in brain tissue and look to see between the axon of one neuron and the dendrite of the other. Is that a continuous flow, or is there a space? That is on the nanometer scale of resolution, and this just shows the axon, perhaps more detailed view of the axon of this neuron connecting to the dendrite of the next neuron. And here is an electron microscopic picture, EM picture of a synapse. This is at a resolution of 0.2 nanometers. So what this is doing is taking, let's say, this is a synapse right here, okay? We are taking a section through the axon and the dendrite and looking at the cross-section in this picture right here. Here is the dendrite. On the dendrite was called a little spine. It's like a little mushroom out onto the surface. And onto these spines, axons terminate. AT is axon terminal. And what you can see in a fuzzy way is these little circles. These little circles are synaptic vesicles. They're filled with neurotransmitters ready to release at the axon terminal. We're going to be focusing on that in 2 lectures from now. But for this one, for the structural lecture, the key point is this space right here. There is a blank space. Here is a membrane. It's the end of the axon terminal, and there is a space right here between the axon terminal and the dendrite, the dendritic spine right here. Again, it took about 50 years for us to get this resolution to 0.2 nanometers, be able to show definitively that there are separations, physical separations between one neuron and the next. Okay. More details on neurons and glia. This comes from your book. Again, just nicer pictures of the different major components of neurons. Here is a picture of the cell body with the dendrites coming out around. You'll notice there's lots and lots of dendrites, typically only one axon coming out. Typically, it's much thinner than the dendrites. Another important component of the neuron is called the axon hillock. The axon hillock is right here, right as the cell body turns into the axon. This is a very, very important location in the cell in terms of integrating input, and it decides whether the cell is going to fire action potentials down the axon or not. We're going to talk more about the axon hillock, but for this lecture, I just want you to know where it is on the cell. The axon hillock is located just where the cell body turns into the axon. Okay, the output structure. Dendrites, as I said, receive input signals. You can see it has a huge space onto which inputs can be deposited onto the cell, and one long axon that terminates in particular locations. There's one axon, but there can be many branches at this level. So it's not like one cell body only contacts one other neuron. It can contact many hundreds of neurons. And this output is transmitted to other sides. And importantly, as Cajal first deduced from his anatomical drawings, there is a specific unidirectional and non-backward going direction of information flow from the dendrites down to the axons. Okay, what are some unique components of the neurons? We talked about dendrites, the input zones. They receive inputs from more than hundreds of other neurons, and there are many dendrites per cell. Axons carries the output. Usually there's one axon, and it transports, the axons are transporting chemicals from the cell body to the terminals. It transmits electrical impulses. That's going to be the focus of Monday's lecture, to the terminals, and the speed of that transmission is determined by size, by myelin. We talked about what makes the white matter white. It's myelin. It's a fatty coating of insulation, and sends outputs to again lots of different other neurons. This is a table from your book. You should know all these distinctions, the differences in number, many individual dendrites, usually one major axon with lots of terminal branches off of one major axon. This is dendrites taper progressively towards the end, so they're very thick when they're near the cell body and get very, very thin. And axons are uniform distance. They are thinner than the thickest part of the dendrites. Axon hillock is in the axon. It's not in the dendrite. Covering or sheathing. Axons are usually, but not always, covered with myelin. We'll talk about why that is the case and what that means in terms of what the axon, what that neuron would be able to do when it's either covered with myelin, not covered with myelin. Dendrites never have myelin on them. Length. Axons could be really short to several meters long, and dendrites are usually much shorter than axons, as we talked about in that drawing of the retina from Cajal. Branching, axons usually branch along the length, and branches tend to be perpendicular to the main branch, and dendrites branch along the length, and branches occur over a wide range of area. Okay. So let's talk about the range of different sizes and lengths of neurons across the animal kingdom. Flies have nervous systems. They have neurons, and they work really well. How many of you have not been able to catch a darn fly when he's buzzing around your head? Yes. Okay, it's really annoying. His neurons can work faster than yours. And we have this all the way compared to the giraffe example that I talked about, where his motor cortex neurons are going all the way down to get all the way down to move his little hoof. Okay? So a wide range, and it's really amazing to think about how widely the size of these neurons differ across nature. I love this picture from the book. I recommend that you go and study it. Just to appreciate the different sizes. All these neurons are drawn to the same scale, where this scale bar is 200 micrometers. And you can see the different sizes and the different shapes of different neurons from different structures. This huge motor neuron comes from the lowly locus. Of course, this is much blown up, but you can compare this huge neuron in the locus, where a major thing is movement, to this neuron in the monkey visual cortex, doing major computations, not that big compared to the locus neuron, but also very important computationally. We have zebrafish here, rat thalamic neuron, turtle neuron, tree shrew, a tiny little monkey, New World monkey. Here's a mouse neuron from the globus pallidus, part of the striatal system. Here is a human retinal ganglia cell that we'll talk a lot about in the vision lectures. Here is a pigeon tectum ganglion cell and a pigeon tectum pyramidal cell. So you can see, oh, here's another monkey cell. So you can see that there are widespreading projections of these cells, but it's just amazing to see how big an individual locus cell can be and how small the monkey cells can be, even though their nervous systems are such different sizes. Okay, so 3 common shapes of neurons that you should be familiar with. This is one of the review questions. 3 major neuron types. Even though I should say that there are hundreds of different kind of categories of neurons, these 3 shapes are the most common. The most common of the 3 are called the multipolar neuron. Multipolar because it has many different dendrites coming off of the cell body. These kinds of neurons are common in cortex. We'll be talking about pyramidal cells in the hippocampus and in the cortex. That is a subdivision of the multipolar neuron. Bipolar neurons are called bipolar for a reason. They have a dendrite going out here and an axon coming out from the other side. They're just kind of skinnier versions of the multipolar neuron, and these are common in sensory systems. For example, in the retina, there are bipolar cells, a bipolar cell layer in the retina. And finally, the third category is one that we've also talked about and mentioned before, even though we haven't gone over the morphology. It's called a monopolar neuron. Here, a cell body, one process comes off the cell body, and then it splits into the dendrites on one end and the axons on the other. And these cells are the category of cells that transmit info from the body into the spinal cord. Now, do you remember what we talked about? There's a special compilation or grouping of cells near the spinal cord on the dorsal end of the spinal cord. Anybody remember what that's called? What is a grouping of cells called? Ganglion. So what is that grouping called on the dorsal part of the spine? Dorsal root ganglion, perfect. And so these are the types of cells that are in that dorsal root ganglion. Those ganglion cells, those cell bodies, sit in that sac that I pointed to, the kind of outcropping, dorsal root ganglion. And then their cell bodies are going out, the dendrites are going out to process information, for example, in the sensory system. Their dendrites are here in my fingertips. And then that dendritic information is bringing that sensory information in, and then it goes back out to the axons, and this goes into the spinal cord and up through the somatosensory processing area. Okay? So 3 common types of neuron shape: multipolar, bipolar, and monopolar. Okay, so pyramidal cells, a category of the multipolar neuron, are very common in the neocortex, okay? And so here, what I'm showing is simply a thin section through the cortex, and what it's stained with is it's called a basophilic stain that only stains the cell body. So we can see approximately the cell body size and shape, but none of the processes, the dendrites, the stain is not high enough resolution, or it's not small enough to get into the dendrites and the axons. So we're basically looking at cell body stains. And then each one of these individual cell bodies, if you would stain it with with a Golgi stain, you would see that it has these huge axons that go up this way. This is layer 1. This is the outside of the brain. The outside of the brain, the outer covering, corresponds to what's called the first layer of cortex. And as you go down towards the white matter, that is the axons, you have typically 6 different layers. I should say that layer 1 is cell-free. These are where all the axons are running. So that means that the covering of the brain is made up of axons that are running across the surface. Layer 2, small cells, very small, called granule cells. They're just small and round. Layer 3 cells tend to be a little bit bigger than layer 2 cells. Layer 4 cells can differ. These are also a form of granule cells. And the deeper cells tend to be bigger. There's a layer 5 and a layer 6. But these 6 layers are typically stained with what's called a Nissl stain that's specific for cell bodies. And you'll be looking at some Nissl stains and possibly some Golgi stains, I think, in the microscopy lab in a few weeks. Now, what this is a much larger section through one part of the monkey brain. And here is what I show you this because this is one of the most easy to see differentiations between 2 cortical areas. The art of differentiating between one cortical area and another just based on a Nissl stain is called cytoarchitectonics, which is the study of cell-body cytoarchitecture with the Nissl stain in the cortex. Okay? So here is one region of cortex. And you can see there's a layer 1 here, no cells. Layer 2, layer 3. Layer 4 is very, very thick here. It has 4A, 4B, 4C. Layer 5 has fewer cells, and layer 6 is very thick. And you can see that right here, this very thick layer 4 with these 4 subdivisions disappears. This is the differentiation, and this is the boundary between area V1, primary visual cortex. Why does it have such a strong, thick and differentiated layer 4? Because this is where all the thalamic input, from thalamic visual input, inputs to the cortex. And so this is a huge input area. This is a transition between primary visual cortex and V2, or secondary visual cortex. No or much less thalamic input into V2, and so it loses this very thick and differentiated layer 4. So this is just to give you an example of how you differentiate between these different areas of cortex using a very common stain used since the 1800s, the Nissl stain. We still use it today in the lab. Let me just give you a quick overview of what people see that's different in these cortical areas. Motor cortex, primary motor cortex, those brain areas that are going down and moving your toe, going all the way down. You'd think they'd have pretty kind of strong cells, the big cells that go all the way down. In fact, they do. Can you see? I know that it's hard from the audience back there, but can you see these bigger, darker spots right here in layer 5? Those are probably the largest neurons in the human cortex. These are primary motor neurons in primary motor cortex. Those are the ones that are going out and controlling motor functions throughout the body. Okay? You can see how different this is and the lack of these big old cells in association cortex, higher levels of cortex. And finally, this differentiates from primary visual cortex, and you can see this thick layer 4, this highly differentiated layer 4 right here. Okay? So I say this for a reason. It goes back to the history of studying which brain areas are important for what. We talked about phrenology that said, well, I'm not interested so much in the brain. I'm just going to look at the bumps in the head. That didn't turn out to be so useful. But from the late 1800s, early 1900s, again, during the same time that Cajal was making his historic observations, many other neuroanatomists were using stains exactly like this. We're using this stain, the Nissl stain, to try and look and differentiate different parts of the cortex just based on how the cells were aligned. And this gives you a feeling for all the different subdivisions that different people came up with. Smith in 1907 had subdivisions like this. He actually didn't even use the Nissl stain. I lied. He took thin sections and simply looked at them in the light and looked at how dense the light came through. And he said, okay, well, I have clear differentiation in these areas. Brodmann did use the Nissl stain. He looked at human brains. And let me just tell you, if you cut the human brain, you saw how big it was. If you cut it in 50 micron sections, that's a typical thickness that you cut the brain in. Imagine how many sections you have to look at to characterize the entire human brain. These guys were crazy. They sat in their lab and they just looked at human brains, and not only human brains, monkey brains, sheep brains, rat brains, flying fox brains, and tried to glean the kind of principles of cytoarchitectonic organization across these different brain areas and glean the function of or differentiation of brain areas and perhaps the function through cytoarchitectonics. This is another beautiful one, von Economo. It's a black and white drawing, but the book that he published in 1929 was color. It was actually beautiful, and his descriptions were absolutely precise and easy to follow. I know because I read them as I was trying to discover and characterize one part of the brain that hadn't been studied so much. And comparing Brodmann and von Economo, Brodmann's descriptions were very, very vague, hard to follow. Von Economo's were very, very precise. But today it's Brodmann's areas that are used in fMRI studies, in lots of different studies to characterize the cortex. And I've always been puzzled why Brodmann ended up with the higher, all the fame and fortune, and von Economo did not. And my conclusion is that he had better PR. He had easy to understand. They were just Brodmann areas, 1, 2, 3, 4, 5, 6, 7, and 9, 10. Easy to understand. These were much more complicated, even though the descriptions were better. And so it's Brodmann that we use, even though I have to say that if you go back and read his primary work, it's not nearly as detailed and beautiful as von Economo. Okay, so now let's go into glia cells. Second major component. So glia cells outnumber neurons 10 to 1. Okay, they outnumber neurons 10 to 1, but there are many, many, many different, a larger category of different neurons, shapes, sizes. There are hundreds of different neuron types and only a very small number for glia types. What do glia do? First, glia means glue. So early neuroanatomists thought that glia actually structurally held the brain together into all those folds. That turns out not to be the case. They do a wide range of functions, and I was mentioning they do even more than we appreciated, and that is a very, very hot area of research right now in neuroscience. One, they communicate with each other and with neurons. And we thought that they did kind of just support functions. They're just there to support the neurons. But in fact, more and more research has been suggesting that they're doing some computations similar to neurons as well. And they are not just support, but they're critical support for the computations that these specialized neurons are doing. They provide raw materials and chemical signals that we'll talk about in a second. They provide protection for neurons. For example, there are glia cells that form kind of a barrier around a particular synapse, not to let other inputs in and perhaps to sheath that particular synapse so that the neurotransmitters aren't flowing out and flowing in. They can form kind of a physical boundary. And they do damage control. If there's damage in the brain, for whatever reason, they can phagocytose the damage coming up. And as part of that damage control, they do a lot of the debris removal if a cell dies or, again, if there's damage. Okay, 4 major types of glia cells. You need to know all these major types. First, astrocytes. Astrocytes nourish and support and scavenge at the synapses. We're going to be looking at individual examples of these. Microglia, as the name suggests, they're much smaller. They multiply at injury sites and seal the area. And they also help to remove the debris. Oligodendrocytes and Schwann cells, both of these make myelin. Sorry, this should be highlighted in blue as well. Oligodendrocytes make myelin in the central nervous system, and Schwann cells are the category of cells that make myelin in the PNS, or the peripheral nervous system. Okay, so 2 of these types of cells are specialized for making myelin. We'll talk about what that means. And 2 of them are support and injury support for cells in various ways. Why should we be interested in glia? Well, the main reason right now that we know the most about is disease states, and disease states involving problems with myelin. Multiple sclerosis is a degeneration of the myelin coating. So we're going to talk a little bit about exactly what myelin does. But if your myelin starts degenerating, you have severe problems in this case, in motor movement. So multiple sclerosis, you get myelin degeneration in the motor system. Glia are also implicated in many disease states. So glia form many of the tumors that arise in the brain. Glioma is a proliferation of glia that could happen if the glia are reproducing at the injury site, or there is simply a tumor, a malfunction of this proliferation function of the glia, and that turns into a brain tumor that can be very dangerous. And astrocytes also, their role is to nourish and support and scavenge, but they also swell in response to injury that can cause the brain edema that happens with brain injury. Brain edema is terrible because what happens with the brain, it's encased in the skull. You swell up too much and you start doing physical damage and shearing damage to the brain itself. Okay, so these are just 3 examples of disease-related aspects associated with glia cells. Okay, astrocytes and microglia. Astrocytes here do some really very important roles. They detect neural activity and regulate adjacent capillaries. Why is that so important? It's so important because neurons really need blood supply to be able to react to what they need to do. And that is why if you have even a temporary slowing of blood to the brain, you can get woozy, you can faint. It's because your neurons need that blood and that oxygenated blood very, very carefully. And what astrocytes are doing is they are monitoring how much the cell is active. And if it starts to really fire a lot and use a lot of energy, what the astrocytes can do is say, hey, capillaries expand, let's get some more blood into this area. A critical function. Microglia, important for all kinds of injury, from severe traumatic brain injury to whatever happens when you bump your head. We talked about you bump the back of your head and you see stars because you're stimulating the primary visual cortex. You're also bruising your brain. And this is also what we talked about in terms of the athletes that tend to put their heads in harm's way a lot with a lot of physical contact. The football players probably have enormous amount of microglia that are proliferating in their brain to help phagocytose the damage and the bruising and the death of the cells that are happening with increased contact and increased concussion. Microglia work to break down debris that forms, especially after damage in the brain. Okay, and so finally, I want to end with the idea of myelin and what it's doing. So we talked about the oligodendrocytes and the Schwann cells, whose role is to make myelin. Myelin is a fatty substance. It looks white when you simply look at the brain, cut it in different subsections. And what these oligodendrocytes do is it produces myelin and it wraps around the axon. So this is the axon cut in cross section. And what you can see here is layer upon layer upon layer of this myelin wrapping, this fatty wrapping. It's just like an insulating substance. It coats long axons are typically coated with myelin, and short axons are bare. Why? Because the major role of myelin is to help synaptic transmission. It is actually helping the electrical activity go faster down the axon, so that when you say, I want to move my toe, you can do it very quickly. Or when a soccer player or a soccer goalie has to actually react to those penalty kicks, they can dive in a very, very fast action. That is all because of the fast conduction that you're able to do down your axons, down your motor cortex to all of your muscles. And that is why if you have damage to either the oligodendrocytes or the Schwann cells making myelin, that you start to have motor problems and slowing and weakness in the muscles that are so characteristic of multiple sclerosis. Okay, so what typically happens is that there are multiple wrappings around a long axon. So a long axon will be all the way down here. You'll have a wrapping of myelin here, and then there'll be a little gap, a wrapping here, a little gap, a little wrapping here. And the gaps, here are two wrappings in a cross section. Here's a gap. So here we're seeing the axon. The axon is bare. And in between these two gaps are wrapping of myelin. This bare gap is an important structure called the node of Ranvier. You need to know this. The node of Ranvier is, again, the blank portion where you go down to the axon. This is where the action potential, those electrical activities, will jump to and then fire off again, and then they'll jump to the next node of Ranvier. And that jumps the allows the action potential to go faster and faster down the axon. We're going to cover this in greater detail, but you do need to know, and I wanted to introduce the concept of the node of Ranvier early here as we're talking about myelin. Okay, and then lastly, here is a picture at a higher magnification of these wrappings of individual kind of layers of this fatty myelin substance. You can see how precise it is and how it's just kind of wrapped with string over and over and over to provide a very thick insulation for the axon. Here you see a mitochondria within the axon, part of those major structures that you see in all cell types. And finally, while the long axons are typically myelinated, not all axons are myelinated. Some are unmyelinated, but they tend to be wrapped around and held together with a sheath of myelin. This sheath of myelin isn't helping electrical conduction, but it just helps them kind of group together. So here is a bunch of unmyelinated axons. So these have slower conductions. These are axons that you need. You don't need as much speed. And then they're all grouped together around, bundled in a sheath of myelin. So two different types of neurons. Okay, so that is all I have today. Next Monday, very, very important. I will say that the next 3 lectures are really going to be the core of this first exam. These are the concepts that you need to know, so I would highly recommend that you all read the chapter on electrical and chemical synaptic transmission before Monday's lecture. You'll get the most out of it. It is dense, and we're kind of going to jump in to the hardcore neuroscience next Monday. So come prepared for that. Have a good weekend.